based on their residence times in the binding site, or equivalently their off rates. Okay, so just quickly, drug target residence time has, over the past 10, 15 years, been increasingly recognized as a key property to optimize. It's not the same as binding affinity, but it complements binding affinity in many cases. For some systems, they correlate well and very well with each other. For other systems, they're not really giving you the same insight. So at least for many systems, KO plays a central role to both drug efficacy, but also selectivity towards different targets. One example could be M3 versus other muscarinic receptors. And also dosage, of course, because if the drug stays bound to the protein for a long time, then you don't need to take a pill that often. Okay. The problem, of course, if you want to simulate unbinding kinetics, is that it's slow. It's based on MD, so of course it's slow. Many of the existing solutions are academic solutions. They are great solutions, but they are also perhaps a bit hard to use for non-experts. So they also rely on quite a bit of expert knowledge and manual setup. So what we wanted to do was to build a tool that could be used quickly and without having to manually choose a lot of settings in a target-specific way. So we have built a new completely physics-based algorithm for scoring ligands based on their activation energy barriers, which I'll show you in a minute. Okay. Our ideas were at the beginning, and well, you're welcome to comment on those, but in order to be useful in early stages of drug discovery, we need a tool that's, first of all, robust. It needs to be able to handle differences between ligands, non-congeneric series. It should also be able to handle cases where the binding modes or the protein conformations are different. If you, for instance, have two or more different protein structures for which different series of ligands bind, then it should be able to capture that. It needs to be fast, because you want to ideally screen many ligands using this approach. So in our case, we were aiming at running end-to-end for a new ligand within a couple of minutes. And accuracy, well, that's always a question. What is accuracy? We want to provide useful enrichment for drug discovery programs, not to provide absolute numbers that are within very close to experimental measurements. So we are aiming at for instance, Spearman and Pearson correlations between our data and the experimental measurements of, well, hopefully about 0.5, but enrichment is more important than absolute accuracy in our view. And of course, it needs to be automated in a way where you can just submit a lot of calculations and get the results back. So, as I'm not a computational biophysicist. I was trained as a quantum chemist, so I naturally took the simple-minded approach and remembered back to high school chemistry the Arrhenius equation. So what we are actually doing in our tool is that we score the ligands based on the lowest energy barrier we can find that connects the bound state to an unbound state. So we generate a handful of trajectories that are close to being minimal energy paths, but they are at least low energy pathways. We can do that very efficiently with our tool. And then we score the system based on the lowest energy barrier we can find. Of course, this is just a potential energy. This is not a free energy. If you put in the free energy, then you get a better estimate, but it's harder to compute. So the solution, and I forgot to put the name in, we call it coffee or K-offy, depending on how explicit you want to be. So it generates a number of non-equilibrium pathways by getting the ligand out of the protein. So you need to start from a bound state. And then you just measure the energy barriers on the way. And you can either then choose to take the lowest energy that you find, or you can make some more advanced Boltzmann weighted sum. These two are often kind of the same, unless you are in a situation where you have multiple degenerate paths or multiple pathways that have close to the same activation barrier. It runs end-to-end in a few minutes on a moderate-sized GPU, like an NVIDIA T4, something like that. You can easily run it on my laptop as well, on a small GPU. Of course, that depends on the protein size, but we are currently just using classical force fields as energy evaluation method. So to translate to a language that's more suitable for this crowd, I guess, the number of energy and force evaluations correspond to what you would usually do when you run 10 to 50 picoseconds of MD. Yeah, and one important thing, we use implicit solvation. So we, of course, that gives us a speed up, but it also allows us to run these calculations without having to equilibrate a large box of water molecules. So we can just plug in your protein PDB, your ligand SDF, and run the calculation just like that. Okay, but the proof of the pudding? Well, we have benchmarked this this approach on basically any benchmark data we could get our hands on, but it's not easy to get good quality public data in this field. But we have been looking at GPCRs, kinases, HSP90, chaperone. That's the biggest and most well-curated data set in this unbinding kinetics field. It was published in 2018. Also DPP4, an enzyme. And we are basically doing quite well. So the take-home message is that these activation energy barriers correlate very well with the residence times that are measured experimentally. And I should note that we have 2 HSP90 bars. One is with the standard tool, which is the one we are almost ready to provide as a product. And this one is a prototype implementation that does some extra refinement on top. But as you can see, we are also able to increase the correlation quite significantly. Yes. Just a little deep dive into some of the data. I know these scatter plots are not as pretty as some of the ones we have been watching over the last couple of days, but these calculations are also much faster. So we started to validate this approach on a couple of small data sets from the literature. We have the M3 receptor and FAK. These are both quite well-prepared data sets. We have a single protein structure. We have a series of congeneric ligands, and these have been docked with respect to a known crystal structure of the natural ligand. And then we have TTK, for which we simply found experimental values in a paper, and then we downloaded the PDBs from the protein data bank and ran directly on those. And they are actually quite different, those structures, as I'll show you, but we still get good performance. So yeah, some more insights. These are all the TTK structures overlaid, aligned, and you can see that particularly there's a loop structure up here that for some of the systems it's very close to the binding site, for others it's very far away. But our approach still enables us to rank these ligands based on their residence times. And the ligands are actually also very different. We have different charges for the ligands for TTK. The percentage of atoms in the maximum common substructure is only 19%, so they are basically not similar, while the others are. But in both cases, we are able to get a good correlation. For the HSP90 dataset, well, again, not as pretty as other scatter plots we've seen, but you do see some signal. You do get some enrichment if you choose the best ones based on these calculations. And these are, again, 1 to 2 minutes per data point. And then if we go to our prototype algorithm, we get much better correlation. And I should say that we have these correlations are reported without removing any outliers from the data set. If you go back to the original publication of this data set, they get a very good correlation, but they have also removed 15 out of 70 data points for being outliers. Yes, and then finally, we also tried to ask the inverse question. Can you use this approach to rank different mutants of the same protein for a given ligand? And we don't have much data to work with here. So So we have been looking at the M3 receptor with tiotropium, a known very long residence time binder. And then we introduced a set of point mutations close to the binding site, and we had some experimental values that we could compare to. And of course, the correlation is not perfect, but it seems to show some correlation between what we predict and the experimental K-off values. So hopefully, when we have tested this quite a bit more, if we get the data, we would love to be able to see if this can be applied to, for instance, something like target selectivity of small molecules, especially because selectivity can in many cases be kinetically induced. So it's a relevant question. Okay. Well, can I try this at home? Well, you can soon. So we are actively seeking strategic partners to test this tool and try it on their own private data. We don't need you to send us data. We are developing a tool, an on-premise tool that you can install locally and try, and it should be ready within 1 or 2 weeks. It should run on basically any Linux computer with a GPU. It can run on a CPU as well, but it will be much slower, as you know, CPU versus GPU with MD codes. But if you're interested, you are very welcome to reach out. And then just because we're at the free energy workshop. Our tool not only generates a score, it generates a pathway. So can we extract anything more from that pathway than just a number? Yeah, well, yes, we can. Of course, you could think of many, you could think of extracting fingerprints, interaction fingerprints between the protein and ligand to identify relevant parts of the protein and ligand that interact strongly and give you a long or short residence time. But you can also start using these pathways as starting points for enhanced sampling. So in this case, we just took the easiest old-fashioned approach, implemented umbrella sampling, used the sample 6 octa-acid systems, so very small post-guess systems, and we ran umbrella sampling on those starting from our unbinding pathways. Less than 40 nanoseconds of simulation per ligand. I know 40 nanoseconds is a lot, but it's much less than what was otherwise reported in the sample 6 review. And we get ranking performance on par with the best ones in the challenge. Of course, the RMSE is larger than what people usually prefer, but this is also, I guess, to a large extent due to the choice of implicit salvation. But this is just an example. You could take these pathways and you could put any enhanced sampling method on top. You could use milestoning. You could use them for metadynamics, for computing both binding free energies, but also absolute kinetic rates. So that's something we will actively pursue in the future. And we also did some alchemical stuff. So just to try it. This is a single decoupling approach. So again, implicit salvation. We took the same systems, and then we turned off all the interactions, also the ones mediated through the implicit solvent model. We get performance that is on par with the umbrella sampling, and it's the same cost. So just to show that we've been playing with alchemical methods as well. But that's not our main product today. That's all for me. Thank you.
Okay, we have time for a couple questions.
Yes, in principle you do. We haven't really benchmarked it thoroughly for these simulations or these results I showed you. We only run 3 per system, but in many cases you see that if you run more, then you are more likely to find one with a lower activation energy. But you, of course, if you are shifting your points downwards, then you need to shift them all downwards to get the same correlation if the correlation is good with few data points. So it's a balance, but in principle, yes, there should be some sort of convergence if you run enough. But we haven't benchmarked it thoroughly enough at this point. a good question. So yes, of course, if we put too much work into the system, then we can just skip some barriers and not get the resolution we want. So we need to do it in a very balanced way. But sure, you need to know how to set your parameters. But all these simulations that I've showed have been run with exactly the same parameters. So there's no target-specific tuning of parameters that you need to do. You don't need to run a long MD simulation first to see which parameters make your system more or less stable or something like that. That's what you would usually do with random acceleration MD. Even though you only have one parameter, the force constant, then you need to make sure that you set it correctly for the given protein ligand system.
Okay, let's thank our speaker.